All right. Hi, everybody. Thank you for joining us for today's SLAM seminar. I'm delighted to introduce today's speaker, who's Dr. Emiliano Perez Ipiña. He is a postdoctoral scholar right here at Johns Hopkins, where he works in Brian Camley's group, doing theoretical studies of motility and migration in microorganisms. And previously, he got his Ph.D. at the University of Buenos Aires. So, Emiliano, take it away.
Okay, thank you very much. Thank you for the nice introduction. And I would like also to start by thanking the organizers by giving me this nice opportunity to present my work here. Can you hear me okay? Okay, so today I'm going to talk about some motility, and in particular about how cells interact with the environment to change or modify their motility behavior or their migratory behavior. Sorry about that. So this talk is going to be to have two parts. The first part is going to be about how cells may use a footprint to guide their own motion. And the second part is going to be about something new I've been working on that is going to be about how cells navigate through environments where they have multiple guiding signals. Okay. So why cell motility? So cell motility is essential for many different fundamental living processes. So here we can see, for example, so we have the case of wound healing, where cells are going to move collectively to close a wound. It's very important during development, where we can see this very nice synchronized and orchestrated movement of all the cells to create the different regions and parts of the embryo. Cell motility also plays a very important role during for our immune systems. Basically, here we can see a neutrophil cell chasing a bacteria. And also, cell motility plays an important role during cancer and cancer metastasis, where we can see how cells start moving out from this tumor to start invading new tissues. But cells, they don't do this alone. They don't live isolated. They live in complex and very changing environments. As we can see here on this cell that is moving over the extracellular matrix. So we can see that this cell, as it moves, is going to pull and push from the fibers, creating local deformations. Not only that, but cells can also degrade extracellular matrix and create new extracellular matrix. So what we see is that there is this interesting close relationship between cell motility and environment, where cells, as they move, they're going to change their environment. So, for example, changing the topology, the density of the stiffness of the fibers. And these changes on the environment can change or affect the way cells behave. So that this poses an interesting question, that is how cells may use the environment to control and modify their own motility behavior. So this brings me to the first part of this talk, where I'm going to talk about how cells use footprint to modify their own, to guide their own motility. And this work has been inspired by the experiments done by Joseph D'Alessandro and Benoit Roux, where they create these linear straps that were coated with fibronectin. So fibronectin is basically like a sticky glue where cells are going to adhere. So basically cells can only move over the regions where fibronectin is, and they cannot escape those regions. So they fabricate these linear tracks, and they place MVCK epithelial cells, and they just observe the cells for a long time. And what they found was that these cells were secreting or leaving a footprint over the substrate, and they were using this footprint to create this oscillatory motion with increasing amplitudes. So the way it works is that these cells, where they are moving or crawling over the past footprint, they are going to tend to move persistently. But when they reach a region where there is no footprint, so that means a region where the cell hasn't been yet, now this cell is going to stop and turn around and start moving towards the other direction. So basically, this cell is just going to be bouncing between the two edges of the footprint. The increase in amplitude can be understood because every time that the cell hits one of the edges of the footprint, is going to extend it a bit longer. So we wanted to understand how cells can use the footprint to produce this type of oscillations. We also asked ourselves, like, okay, so maybe they can use footprint to create other type of motility behaviors. And if so, maybe cells can control this. In a more abruptly view, we ask what is the role of cellular footprint in cell migration. So to try to address these questions, we basically build a mathematical model where cells basically move, and as they move, they're going to leave a footprint over the substrate and interact with the footprint to change their motility behavior. So this model So to model the shape and motility of the cell, we use the phase field approach, where basically the cell is described by a field that takes the value 1 in those regions where the cell is, and 0 in those regions where there is no cell. And then the cell boundary is given by where the field takes the value 1/2. And then the field is going to evolve following this equation over here. So this equation has 2 terms. And the first term is the one that is driving the motility. And in particular, the motility is driven by this rho function that describes the concentration or density of a polarity protein that we call RAC. So essentially what is happening is that this cell is protruding or extending in those regions where rho is high. And then the second thing that we ask, the second term basically is the minimization energy function, where we are going to ask the cell to minimize this double well potential. So basically, this is what is defining inside and outside of the cell. And then we also ask the cell to minimize the linear tension on the membrane. It's important to note that here we are not asking the cell to minimize a volume, so cells can change the volume in our model. So as I said, the motility of the cell is driven by this polarity protein that we call RAC. So RAC can be in 2 states, can be in an active state that is going to be bound to the membrane, and it's going to accumulate at the front, creating a polarity front that is going to drive the directionality of the cell. It can be also, sorry about that, in an active state that is going to be distributed all over around the cytosol. And the dynamic of the active RAC is follow this equation over here, where we can see that RAC diffuse over the membrane and can also convert between active and inactive, given by this term here. And what is important over here is this term that describes a positive feedback mechanism. That is what is going to guarantee the accumulation of active RAC at the front. So basically, when we have some accumulation of active RAC at the front, that is going to trigger more accumulation of active RAC. And in that sense, the cell is going to be able to sustain a polarity front and move persistently over time. So for the footprint dynamic, we also follow a simple scheme over here. So basically, we assume.
Sorry, Emiliano, can I ask a question on the previous slide? Is there some conservation of total row plus row site? Or are they totally different populations?
No, there is a conservation, and also we are. This is something I didn't mention, but we assume that the dynamic of the inactive RAC is much faster than the dynamic of the active RAC. So basically, we can make a quasi-stationary approximation and basically assume that this cytosolic RAC converges rapidly to equilibrium in the cytosol.
Okay, thanks.
Yeah, thank you for the question. Regarding the footprint dynamic, I was saying that we assume a very simple case here where the footprint is going to be deposited under the cell surface and with a given secretion or deposit rate. And we also assume that the footprint can degrade over time. And also we are assuming that the cell doesn't interact directly with the footprint, but instead we assume that there will be some type of receptors over the surface that are going to bind the footprint molecules, like could be, for example, integrins. So basically we assume that the footprint is going to respond following so the cell is going to respond to the footprint following this equation over here. That is kind of like a Hill equation. And here we can see the response of the footprint as a function of the footprint concentration. So basically we can see this sharp transition and this function acting like an on-off function. So lastly, we need to say how the footprint changed the motility behavior of the cell. For that, we assume that the footprint is kind of entering here, basically controlling the positive feedback term. In the equation, we can see it over here. So basically, the way this works is when the cell is crawling over a region where there is a footprint, now this positive feedback mechanism is going to be on. So that means that the cell is going to be able to sustain a polarity at the front and it's going to be able to keep moving in the direction it was coming from. But now if the cell reaches a region where there is no footprint, now this positive feedback is going to be off and it's not going to be working. So that means that the cell now cannot sustain a polarity and that eventually the cell will stop. So let me now show you some simulations of our
Sorry,
model.
I have a quick question, Emiliano.
Yes.
Can you so the variables in this description are the dynamical variables are rho. So because rho cytosol you said was dictated quasi-statically by rho, and then there's the footprint, and how do you obtain the center of mass velocity, and how do you obtain the size, and what's the and do you have any description for the cytoskeleton?
So no, we don't describe the cytoskeleton here. So we don't measure the center of mass velocity of the cell. Basically, what we are doing is we are using this phase field model. I think everything is going to be more clear when you see the simulation. But basically, what we have is a field that is describing the cell shape and motion, and this field is basically evolving because extend the membrane in those regions where the concentration of this active rock is large. And then it has to minimize an energy. So basically that means that it has some constraint in the tension of the membrane. So that is basically what is going to define how the cell evolve and the shape of the cell.
Yeah. So but the phase field primarily tracks shape. What I'm trying to understand is because the gradients of rho are setting up a polarity in the cell, and so there seems no obvious mechanism of being advected in the direction of that polarity, which I would imagine would be present because in
I think I understand what you mean, and yes, we don't have an advective force here. So basically, that force that is driving the motility of the cell is given by again this term over here. So basically, the role that is playing the active wrap is basically over here. So what it's doing is basically in those regions where the active wrap is high, the cell extend the area. And I didn't mention, but we have this beta that is basically control the retraction of the cell. So basically there is a balance. So this is what is protruding the cell. This is what is the retraction of the cell. And all this is happening at the level of the membrane. So there is no force that is being applied on the center of mass of these cells.
Okay. And there's a question in the chat regarding if F cell is the force due to the substrate on the cell.
Oh, this F cell? No, this F cell, oh, maybe it wasn't the best symbol to use. No, this is just the footprint. So F comes from footprint, not force.
Thank you.
Yeah, not the best choice of letters. Okay, so I think everything is going to be much clearer when I show you some simulations. So we start by simulating or mimicking the experiment. So we have our linear track and we place the cell in the middle, and then we observe how the cell evolves. So here on the right, we have, on the left, sorry, we have the cell that is described by the purple boundary, and the flickering thing on the front of the cell is basically the distribution of the rho. So we can see how this cell is kind of oscillating, and we can see how these oscillations increase their amplitude over time. And here on the right, we are seeing the footprint. So basically, we can see how the cell lay down the footprint all over those regions where it has been before. Now we'll see that once that it gets to the edge, it's going to extend the footprint a bit longer and then turn around and start moving to the other direction. So basically, we were able to reproduce the observations from the experiment, having this oscillatory motion with increasing amplitudes. So then we wanted to use our model to kind of explore a bit more by changing some of the model parameters. It came out that one of the important parameters here is the footprint secretion rate. So basically, it's very important how fast the cells are able to secrete or lay down this footprint. So here we are going to see a simulation in which this secretion rate has been increased. So now we see that the cell, instead of oscillating, now the cell moves persistently, and move persistently even into those regions where it hasn't been before, where there is no previous footprint. And the reason why this is happening is because this cell is basically secreting so much footprint that is basically being able to build the necessary substrate to keep polarized and moving persistently. And we have a cartoon that kind of explains this in a very clear way. So we see that we have this transition between oscillatory motion to persistent motion that is controlled basically by the speed at which the cell lay down the footprint. It can be also controlled by this parameter KT that is basically defining at which concentration the cell is going to respond, or basically the concentration that the cell needs to sustain the polarity at the front. So we can make this phase diagram of the transitions, basically where we have in this area oscillations, and in this area the persistent motion, and then we can see the transition happening over this region. And I highlight this this line over here that basically estimate where the transition is going to happen, and that is happening where the secretion rate is proportional to this KT parameter. And I will come back to this in a few moments later. So so far I show you in those simulations that the footprint was not degrading over time, and that was based because of the experiment. They observed that the footprint basically don't degrade during the experimental time. But now we wanted to use our model to explore a bit more, and now we consider what would happen in the case that we have degradation. So again, we have the cell over here, and we can see our cell that starts oscillating. But now we don't see that the amplitudes increase over time, but we see that the cell reach to a maximum amplitude and remain over there, remain oscillating with that amplitude. We can understand that if we take a look to what's going on with the footprint. So now when the cell tries to go back to the previous edge, it finds out that the footprint is no longer there, so it has to rebuild it. So basically, this degradation of the footprint set up a maximum distance that the cell can go away and come back. And in other words, this degradation of the footprint basically define a confinement region for the cells. So we can plot the amplitude as a function of time, and we can see how the cells reach these steady-state amplitudes. And we can see also how these steady-state amplitudes change with the degradation rate. So the higher the degradation rate, the smaller are going to be the oscillations, or in other words, the more confined the cells are going to be.
Could you remind us, please, how in the equations you get the rho field to turn around to the other end of the cell when it reaches the end of the psi track?
How basically the cell repolarized and changed the moving direction?
Right.
So we have to come back to this. Okay. We have to come back to this. So this is the footprint over here. And this footprint basically controls the activity of RAC. So if we have footprint, that means that the cell can remain polarized. But the moment that there is no footprint, this repolarization term is not going, this positive feedback term is not going to be working. So, here. So the moment that the cell start on the tip of the cell, start seeing that there is no footprint, those areas of the cell are not going to be able to sustain active RAC at the front. So basically, there is going to lose the polarity over there. And that is going to happen slowly. Basically, what you can see from the simulation, that is what is going on is that the cell kind of shrinks when it reaches the edge, and at some point lose the polarity. Since there is more footprint on the other side, it's going to repolarize in the opposite direction and keep moving. Okay.
I have a kind of related question. I think even in the case where the oscillations weren't happening because it was laying down more track, it looked like the cell shape was still going through these oscillations. Like every once in a while, it would, the RAC would sort of, or the active RAC would sort of, spread like here, spread out through the cell, and then it would do that again. Is that something you feel like is sort of an artifact of the specific form of the reaction diffusion equation that you've chosen, or do you think it's a real thing and it would happen in experiments too?
I think it's a good question. I would say that most probably is something that is coming out from our model. So basically what's going on is there is kind of a critical size where if the cell is too small, basically it's not going to be able to sustain this polarity. So basically what's behind this is that RAC is solving like a bistable system. So the RAC is going to be, the cell is going to be polarized if we can stay in that bistable system where we have a front that is polarized and the back that is not polarized. But if the cell volume is very small, now the cell cannot, so the solution of the RAC inside the cell cannot sustain this kind of bistable equilibrium. So basically that means that the cell loses the polarity. So what is going on is basically that if you see that cell is slowly shrinking, at some point it gets a moment that is too small and it loses the polarity. And the moment that happens, it creates kind of two fronts, and that is going to elongate again the cell. So most probably this is kind of like an artifact of the way this model is working. But that doesn't mean that it cannot be observed in real systems.
Can I ask a related question over here? So in the phase field, what because the dynamics you have for the phase field was non-conserved, how do you impose some sort of area conservation, or is there a minimum? How do you enforce the minimum size the cell can reach? And there seems to be the cell doesn't stretch arbitrarily, nor does it collapse to be very small. So what in the phi dynamics is preventing that?
So as you said, we don't constrain the volume of the cell or the area of the cell in this case. What is defining the minimum size of the cell is basically the amount of rack that we have inside the cell. And also the shape of the cell. So here we have a more innovative cell. So that's going also to change that. Basically, how much the cell can elongate, that depends on our constraining the elastic energy that the membrane can have. So, because that is something that we are asking the cell to conserve or to minimize, basically.
So then this sort of so the dynamics of rack, the rho field then becomes quite important, because it has to turn over fast enough that it prevents. It needs to be faster than the dynamics of phi, so that phi doesn't collapse, and you have sufficient amount of rho in the inside to allow for extension. Is that?
I think that's right. So the dynamic of rho is faster than the dynamic of the evolution of the field. Okay. And also I think that makes sense, because cells actually move pretty slowly, and this reaction inside the cell they happen in a much faster time scale than actually the movement happens.
Sure. Yes. Okay. Thank you.
Okay. Where were we? Okay, so we were saying that basically by having degradation we can confine cells. So we wanted to get a bit more insight of what is going on with the model. And the phase field model, as we have been discussing, is a bit quite complicated to understand and to get analytical results. So we tried to build a simple model, in this case a 1D lattice model, where we have a cell that is going to follow very simple deterministic rules. So this cell at this lattice position is going to ask the question, is the footprint larger than 0.5? And if the answer is yes, then the cell is going to move towards the next lattice side following the direction it was coming. If the answer is no, then the cell is going to switch direction and start moving on the other direction. So in order to give you an example of how this works, I built this example where basically we have a cell over this substrate. So this substrate is basically covered with a footprint that is equal to 1 all over this region that is covered on red, and is 0 otherwise. So if we place the cell and start asking what the cell is going to do, so the cell at time 0, if you ask the question, is the footprint larger than 0.5? Then the answer is going to be yes, it's going to move to the right. And it's going to keep asking the same question, and the answer is always going to be the same until you reach this edge over here where there is no footprint. So at this point, then the cell is going to switch direction and start moving over this side. And since there is footprint here, it's just going to keep moving straight until it hits the other edge. So basically, this cell is just been bouncing between these two edges. But of course, we also want to consider that the cell is able to secrete their own footprint, and that the footprint degrades over time. And so now we can use this model to kind of ask what is the condition the cell has to fulfill in order to see these state oscillations. The condition should be that the amount of footprint that the cell secretes at the edge has to be equal to the amount of footprint that degrades on the edge during the time that the cell takes to go to the other end and come back. So this is what we wrote over here. And now we have this expression from where we can derive the value of the steady state amplitude. So we finally get the expression over here. And even though that our model was pretty simple, gave us this expression that is actually very powerful to reproduce or predict some of the prediction that we made with the more complicated phase field model. So basically, we can reproduce how the steady-state amplitude depends on the degradation rate. And not only that, but the model also allows to predict when the transition from oscillation to persistent motion is going to happen. So basically, this is given by this denominator over here. So basically, when this goes is equal to 0, the log basically diverges. And this is telling us, okay, this is the moment where the transition to a persistent motion is happening. And from there, we can see that the transition happens when this secretion rate is proportional to this KD value. Okay, so that was the model on 1D over this 1D stripe. So now we want to see how the cells behave if we place them in a 2D surface. So here I'm showing you a low secretion rate. So this is the analogous case of the oscillations in 1D. But now cells, instead of oscillating, they are going to move in circles. And if we move forward the movie, we can see how this circles expands over time. So this is similar to what happened in the cells in 1D, where the oscillations increase their amplitude over time. So these circles are expanding over time. But now, if we go to a case of a high deposition rate, or basically a deposition rate that is above the transition, what we find is that the cell is able to explore the space, but something interesting happened when the cell cross over the previous footprint. So now the cell, instead of keep moving in the direction it's coming, is going to turn around and start moving over the previous footprint. And not only that, now the cell is kind of trapped doing this loop over the footprint, but in this turn, it decides to go to a new direction and start exploring a new space. So we have this kind of really interesting behavior where the cell is able to explore the space, but at the same time has this slide or loosely confinement, where it has a preference to move over previous trajectories.
There was a question in the chat, was the transition from oscillatory to persistent motion seen in the experiment on 1D tracks? This is back from the 1D section. So was that seen in the experiment, that transition, and by tuning what parameter?
No, I don't think that they have the ability in the experiment to control the parameters as we do, for example, what would be the secretion rate. Actually, in the experiment, they are not actually sure what is the footprint composed by. They have some clue that it can be something that is composed by fibronectin and laminin, but they don't know exactly the molecular composition of this footprint. So they haven't seen this transition. What they have seen is that in some cases they do observe persistent motion. So in many cases they observe this oscillation, but in some cases they also observe some cells behaving in this persistent way. But actually, I was going to come back to the experiment. So I think that question is related to this question. But basically, our model kind of makes some predictions of what is going to happen to this, so we can go back to the experiment and see if this is what is happening. So if we observe cells that oscillate on stripe in 1D, we should expect that in the experiments in 2D, we should be observing expanding circles. And this is actually something that we observe. So this is a trajectory from experiments, and we can see how this cell is moving in an expanding circle. Very nice as we predicted in our model. But this was not the most typical case. It's actually a rare case. Most of the cases, the cells behave in this exploratory way, where we can see some overlap with previous trajectory. So this is more likely to the case of being exploratory, that means cells being above the transition rate, and we should be expecting persistent motion in 1D. So we were a bit puzzled about this. So we went back to our simulations. We basically found that the transition from this persistent motion to oscillatory motion to persistent motion, or the circular motion to exploratory motion in 1D and 2D, they happen at different secretion rate values. So this is interesting because now this allows us to have different combinations of 1D and 2D behavior. So for example, we can have a case where we see cells oscillating in 1D and moving in circles in 2D. So that was our first case. But now there is this region that is kind of close between each other in 1D and 2D, where we can find oscillations in 1D, but exploratory motion in 2D. So coming back to the experiment, the observations on...
Sorry, Emiliano, one second. With regard to the comparison between 1D and 2D, have you all done any, have you all looked at any cases where you vary the confinement so you can interpolate between 1D and 2D?
How would that be?
So,
I'm not...
For instance, by putting soft confining walls or soft potential, say like a quadratic, and you lower the stiffness of that well, so that when it's very stiff, then you're confined essentially to a 1D track. But then as you soften it, then the cell can sort of explore some distance in the y direction. But it's still isotropic, and I wonder if you can use that as to tune the confinement from being to make it soft, and use that to interpolate between the two different sort of regimes that you get.
We haven't thought about that. It's actually, I think it's a cool idea. I'm not sure if you are referring to make this on experiments or make this on simulations.
In the model at the very least, and the experiments, maybe there's a way to do it as well, but at least in the model.
So no, in the model we haven't thought about that. I think it's a cool idea. Might be worth to check it out. On the experience, actually, we don't have control, and
okay.
So we didn't actually participate on the experiments, so we just asked for the data. But it would be really nice if we have some ideas where we can test our model with experiments.
Fantastic, thank you.
Oh, thank you for the question. Okay, so coming back, I would say so having this behavior on the experiment where we see exploratory motion in 2D and oscillatory motion in 1D is suggesting, according to our model, that these cells, the MDCK cells, should be tuned slightly above the transition, so close to the transition point, but slightly above. And also arguing in favor of this idea is the fact that we observe both types of motility behaviors, and that would be consistent with the fact that most of the cells are tuned in the region above the transition point, but because you have some cell-to-cell variability, some of them are going to lay below the transition point, and we should expect this circular motility behavior. So with that, I kind of conclude the first part. So basically, I show you how we can build this motility model where cells interact with the footprint. We were able to see different motility behaviors in 1D and 2D, and we were pretty happy to see that actually those behaviors are observed in experiments, and also that our model suggests that MDCK epithelial cells, they should be tuned very close to the transition, so it is possible to observe these oscillations in 1D and these exploratory motion in 2D, and sometimes these circular trajectories. And I think it's a good moment to ask questions about this before I go into the next part.
Yeah, so maybe for those who are not familiar with the previous experimental work from Benoit Lardou's lab, like what was the experimental proof that it is indeed a footprint that is controlling these oscillations in this? Like, can they, for example, wash out the footprint and then the cell behavior changes? Is that how they demonstrated it?
Okay, so yeah, thank you very much for that question. Yeah, I think it's an important point. Basically, what they found is they made two complementary experiments to this. Basically, one experiment was to kind of fill the tracks with a lot of cells and then remove the cells. And basically, when they place a new cell over there, these cells, instead of moving in a solitary manner, they move like in a persistent way. So basically, with the idea that this substrate were already covered with this conditioned substrate that the cells leave. And then they do another experiment to try to figure out what actually the cells are laying down on the substrate. And what they found is that cells lay down fibronectin, and also they lay down laminin. They actually check because their track they have fibronectin at the beginning, and there is also fibronectin that is secreted by the own cells. So they basically block the ability of cells to lay down fibronectin, and still they observe the oscillation. So basically saying that in between fibronectin and laminin probably was important in producing these oscillations, and not that much the fibronectin.
Can I use this as maybe opportunity to go back to perhaps a question I asked you earlier, which is, so I agree that you haven't included mechanics here, but do you have predictions from the model that you have at the moment for things like traction forces? And what would one, so the question being, how much can you get away with by just focusing on the chemical module setting up the polarity versus for what aspects do you actually need to model the cytoskeletal components and the mechanical components sort of explicitly? And do you have thoughts on that?
I actually not sure if I completely follow the question. As I said, this we don't in this model. So the advantage of the way that we are implementing the phase field model is that we are not following any force or any basically speed on the center of mass of the cell. So all of this is coming from this basically interaction between the activity of RAC basically expanding the surface of the cell.
Yes, no, that I agree. All I mean is that one could refine the model by adding an additional set of variables to describe the concentration of actomyosin, for instance, and that would respond to the local density of RAC and generate stresses, which would dictate an internal flow, which would affect phi, and so on and so forth, making the model more complex. And so the question was, given that you were able to capture a lot of the phenomenology with just the chemical part of the module without including cytoskeletal components explicitly modeled, what aspects of the experiments do you actually need the other bits and pieces, the other complexity that you don't include at the moment? And so are there mechanical observables, for instance, traction forces, that one can measure in the experiment, and can you make predictions using the simple model that you already have? And do they match? Do they not match? And what other observables in the experiments would require you to go beyond the model at the moment as written and include other mechanical components as well?
So I
Maybe
think
there is no answer at the moment, and that's okay. Yeah.
No. Let me just think. I think I can say a few things. So first of all, we know that our model, the way that we implement this interaction with the footprint is kind of probably in the easiest way that we found it possible. But it's not necessarily the only way that you can do it. So you can, for example, make the cell make the footprint, instead of activating RAG, it could be, for example, controlling, for example, the extension of the cell can be controlling maybe the retraction of the cell or something different. So it can be maybe instead of working over active RAG, can be working over myosin or something like that. So there are probably many different ways that you can implement this dynamic between the footprint and the cell motility, given that the complexity and the many different components that takes part in the motility of the cell. I think that if you want to test this idea, yes, you would need to go to the experiments and start probably making some changes in all these components to try to see how they affect the way that cells behave. So, for example, trying to change the myosin, or try to change the RAG activity of the cell to see the way basically that change the interaction with the footprint. And then by having those refined observations, so by having those observations, you can go back to your model and try to make some more refined guesses of what is actually the internal molecular mechanism that is driving this interaction between the cell and the footprint.
Okay, thank you.
You're welcome. So I don't know if I'm going to have enough time to go through the second part. So I don't know what to do basically.
Does an 11-minute version of it exist?
No, I don't think so.
Well, why don't you get us started, and we'll see what we can get through. I'm
I
curious
can try to
about
go a bit fast and try to give you some idea of what I've been trying to do. So again, this is new work. I've been working on this in the last few months, and this was going to be my first time talking about this. I just put together all these slides. So the idea behind this is how cells navigate through environments where they have multiple guiding signals. So usually we study these problems assuming that cells is just measuring one or responding to one of these type of signals, but it makes sense to start thinking of how cells would react in the case that they have multiple signals at the same time. And these multiple signals, they can work together to make a more robust signal for the cells, or they can be conflicting signals telling the cells to do different stuff. So then we ask the question, how do cells integrate all these diverse signals to generate a coherent response? How the cells can prioritize these signals to which one to follow, basically. And one of the examples here is the case of neutrophils, that basically a neutrophil has to follow different multiple chemical gradients to reach to infection sites. So people have been asking how cells do that, and they have performed some experiment where they observe some interesting observation that I'm going to try to go fast over here. Basically, what they observe is that when cells are placed in between two opposing gradients, they will see that cells, instead of going towards the closest source, they are going to move down the gradient towards the furthest or the more distant gradient. And something interesting also happened when they place the cells not in between the two sources, but a bit closer to one source or the other. And they observe basically in one case that cells basically navigate this multi-step manner, so basically going first to one source and to the other. But if they switch the order of the chemoattractants, basically they just see that the cell goes towards one of the sources. And some other experiment has reported cells behave in an oscillatory manner between the two sources. And I say oscillatory like this because actually there is no oscillatory period measured here. So there is no a period of oscillation. So the idea behind this project is how can we make sense of all these observations? People have include to try to explain this, that cells are able to adapt to the chemoattractant, they have positive feedback, or the chemoattractant can inhibit the response to the other chemoattractants. But something that has been missing or hasn't been explored in much detail is the role of how cells sense this chemical gradient in the context of having multiple gradients. So basically what I'm trying to do here is, can we explain all these observations by just considering how cells sense gradients? So I would have to go through how actually cells sense gradients. So basically, they have receptors over the surface that are going to bind the chemoattractant molecules, and then by basically comparing the occupancy of these receptors, cells can estimate the stiffness and orientation of the gradient. But of course, this is a fluctuating process, so the occupancy can change over time. So cells can only estimate this concentration with a given accuracy. And people have been working on this problem, and they have derived equations to estimate this error. This is what I'm presented over here. It's not important to go into detail here, but the only thing that matters is how this error depends in a nonlinear way with the concentration. And the reason that matters is because now if we plot the signal-to-noise ratio, that's basically how well the cells respond to how well the cell can sense the gradient. So basically, we see that there is this non-monotonic behavior where the cell have a maximum of the signal-to-noise ratio at those positions where the concentration is equal to this KD parameter. But if the cell is below or above that position, the signal-to-noise ratio is going to be smaller. So for example, if the cell is far away from the source, it cannot sense it very well. But the same happens if the cell is too close to the cell, then the cell cannot sense very well the gradient, because most of the receptors are occupied or saturated. So by taking into account this kind of interesting dynamic between the concentration and the signal-to-noise ratio, we can understand how cells are able to sense the gradient spatially. So now when we add a second chemoattractant, so we have a source on A and we have a different source on B, so both of the sources secrete a different chemoattractant, and the cell can have independent receptors that bind to this each of these chemoattractants. Now the cell can make local estimation of each gradient independently, and then the cell can combine this estimation to make estimate of the direction it would like to go, and basically by making this vector sum. And these are, of course, subject to fluctuation that depends on how well the cells are able to estimate the local gradient, that depends on the signal-to-noise ratio. So now if we take the mean value of this measurement, we can create these maps that basically tell us what is the average direction that the cell is going to take on each of the positions over the space. So here we have the source A, we have the source B, and all these points, the black curve is telling which is the direction that in average the cell would like to go. So now we can basically place some cells into this landscape and see how they behave, where they go, how they chemotax. So in this case, I'm showing you two sources on A and B. These two sources are exactly the same, are identical, but the only thing that changes is that they secrete a different chemical. And the cells are able to measure both gradient with the same sensitivity. So they have the same number of receptors and the same affinity for both chemoattractants. So if we play the simulation, what we see is that cells or either go to A or either go to B and then remain around A and around B. So this is basically what we call the competing sources. So one of them is going to win, and then the cells are going to stay around those sources. But if we increase lambda, basically lambda is the decay length of the concentration. So larger lambda means that the concentration decays in a longer distance. Now what we see is that now cells don't go towards A or toward B, but they just keep bouncing or in between the two cells. So basically they're confined in between the two cells. So basically here we are demonstrating that cells are able to move down the local gradient and to start moving to the more distant gradient, and that cells kind of oscillate in between the two sources that was observed in the experiments. We can understand this basically if we take a look to the signal-to-noise ratio curve. So basically, we plot the signal-to-noise ratio, and this is the depth, and basically joining both of the sources. So if we place a cell over here, since the signal-to-noise ratio A is larger than signal-to-noise ratio B, now the cells are preferably go towards source A, that is here in this position. The same happens if cells are over this region. So signal-to-noise ratio B is larger than A, so that means cells try to go towards B. But when we start increasing lambda, that means that these branches of the signal-to-noise ratio are start going to move away from the sources. So at some point, these two branches are going to overpass between each other, and we are going to end in a situation like this, that basically now we have that over this region, the signal-to-noise ratio B is larger than A. So in this region now, the cell, instead trying to go towards A, is going to try to go towards B. Similar, this is happening over this region. So basically what's happening is that the cell is kind of confined between this region in the position where both of the signal-to-noise ratio are equal. And of course, if we have that situation, there should be an intermediate situation where basically both of the branches overlap. And in that case, what is going on here is that basically it's measured both signal-to-noise ratio with the same accuracy, meaning that in this case, they cancel each other. So basically, so they don't chemotax over this region here. We can see in this movie here. I'm going to go very fast on this. So basically, we can study how cells disperse over the space, and it's actually pretty cool because we start with a situation where cells are quite confined around the sources. We go through a situation where cells are quite dispersed around in between the two sources, and then again we start having cells confined, but now instead of being confined towards each of the individual sources, they are confined in between the sources. And we have this elongated dispersion of the cells around the source shown in the axis. I'm not going to go into this, but now if we change also the source of the size of the source, we can have a situation again where we confine around the sources, we confine in between the sources, and at some point, everything reverses, and now the anisotropy of the dispersion of the cell change, and now the cells are more elongated in this region. And finally, and this I'm going to be super fast, if we change the sensitivity of the cell to one of the sources, to one of the chemotacting by having more receptors of one type or the other, we can create these hierarchical responses. So for example, here, cell has more receptors of B and A. So in principle, cells are trying to go towards A and B, but if we pay a closer look, what is happening is that cells go to B and remain over B, and cells go to A, but once they arrive to A, they try to go to B. And we can see this in this movie. Basically, we have an uniform distribution of cells, and we can see how cells start going towards A and B, but eventually the cells that went to A are going to start going towards B. So this basically recreate what was observed in the experiments, where we have multiple step navigation of cells that, for example, that start over here, go first to A and then to B, but those cells that start over this region, they just go to B and they don't go to B. And that was basically the end of the second part. Just in exactly. So basically, by considering how cells are able to send chemical gradients, we were able to reproduce some of these interesting observations in the experiments. And not only that, but also in combination with the idea of how the environment can change the behavior of the cell, we can create, in the same way that the footprint can modify the cells more, like more exploratory, more confined, we can have similar results by combining different sources here. So we can have cells more confined or more elongated in different regions of space by just combining different guiding signals. With that, I would like to conclude, and thank you, and thank our group. I will be happy to take some questions.
Okay, well, you in fact had a 12-minute version of that, so exactly what we needed. So thank you very much for that entire talk. Let's give a round of applause. And now we will open the floor to questions. So feel free to unmute and ask your question.
Hi, very good talk. I just want to know in your first part when you talk about the effect of the footprint on the Roche dynamics, you use a Hill function and which is kind of very sharp. So that means that some reinforcement of the footprint by repeating on some tracks or by some other cells on this track to make the footprint higher and higher because it's easily saturated because of the sharpening Hill shape. So it will not have that effect of further accumulation. So I would like to know if you have tried or have some ideas if that's in fact is not that sharp, so that a small, continuous increase of that footprint by repeating on some track, for example, would make some difference. Do you have any ideas on that?
Yeah, you are completely right. So the response of the cell depends a lot on how they respond to the footprint. So the motility behavior of the cell depends a lot on how this responds to the footprint function. And this is a Hill function that basically make the transition very sharp between not responding to the footprint to responding to the footprint. Actually, the Hill coefficient that we are using is just 2, so it's not super high. But we can use a smaller coefficient, and basically that was just going to change. It's just the transition between the responses. So if we make that transition more slowly, what we would see probably is a change in the repolarization dynamics. So it would take more time for the cell to repolarize because now we need to basically lay down more time to lay much more footprint in order to get the right response and be in this saturation phase on the response in the Hill function.
Besides, that's understandable. But besides that, for example, at the intersection of two trajectories, at the intersection point, it's possible that the footprint concentration is higher than the part that without intersection. And then if the cells are kind of sensitive to that difference instead of not sensitive, then it's possible that they might be restricted within that intersection or some behavior else. So like that kind of things.
I think that's I think you make a really interesting point. I think it is true. So it would be I was just thinking in the case of the oscillations in 1D and the 1D stride, probably the dynamic on 2D and how the cells interact with the previous footprint is going to change if we take into account how basically if we make slow down basically the response to the footprint. So basically maybe it can be harder for the cell to change the direction and start moving towards the previous footprint, or it can make it more kind of like an inertia case. So basically the cell slowly try to change the direction, but actually doesn't end changing completely the direction. So basically we have kind of a deflection maybe because of this interaction. I haven't explored this, but it's actually a nice idea and something that it's worth to check. Yeah, thank you.
Thank you.
Okay, other questions? So I wanted to go back to the beginning and at risk of anthropomorphizing the cells, it's clear why they would want to sense multiple gradients. What is their goal in doing these oscillatory motions?
I'm sorry. So you mean like in this case where the cells are?
No, sorry, part 1 of your talk.
Part 1. Okay.
Yeah.
So
right, why would they prefer to turn around at the edge of a place they've previously been to?
I don't know. I think that's kind of like we have to guess why the cells behave in the way that they behave. So first of all, they seem that cells behave in this way. And if I have to speculate why this could be convenient for a cell, I would say, okay, this is a way that cells can remain confined to a region. So basically, by controlling how much footprint they secrete, they can control how much they are confined over space versus how much they explore. So this could be an interesting behavior for cells, like they have some property of exploratory or patrolling some region. So by being confined to one region, because you are basically using your footprint to confine yourself, you are kind of behaving in this more patrol way. So basically you go back and forward, and then you, like, guarantees you that something happened, you will detect it in that way. On contrary, that you just explore the space freely. So I would guess that that could be an advantage, but that's just in the speculation space.
Okay, good. Could it have to do, while we're speculating, could it have to do with cooperation between cells? Could they lay down tracks for each other to follow?
So so it's something I didn't mention. So that's basically some of the where this project is trying to evolve. So basically, how cells use footprint to collectively migrate. So basically, how cells use you can have this guidance mechanism by cells by basically releasing signals to other cells, but you can also use this idea of laying down footprint. So basically, this open the possibility of cells just following other cells by using the footprint. And then the dynamic of this plays an important role, for example, in the case that the duration of the footprint degrades over time or not, basically how well the footprints are defined over space, or if they are not that well defined, it will be harder for us to follow. Basically, I think related to the question of Heisen's Q, basically how the interaction with cells reach this footprint is going to be important. They are going to be able to basically redirect their motion towards the new the previous footprint, or just keep moving. And in case that they jump or they hop on the footprint, are they going to take the right direction or the wrong direction? So there are kind of like a lot of questions that we need to work out. But I think it's a very interesting problem.
Definitely.
One short comment. I just recall that I remember ants. They also can kind of follow the footprint of the other ants so that they can move collectively in a row, something like that, or in a circle, something like that. And if you can do this kind of things with collective cells, multiple cells that can follow the footprint, not just by themselves, but by the others, and then maybe some kind of similar collective behavior might be observed and might be important. I don't know, just
no, I think again, I think these are very cool points. So we have been thinking also like the collective response to the footprint. So basically, people have been studying how cells measure sense gradients when they're in groups. So basically, cells are better sensor of gradients where they work collectively. So you can think that similar phenomena will happen like with the footprint. So if you have more cells together laying down a footprint, that can basically create a stronger footprint because they deposit more time. But there's other thing that still happen, and we are thinking about. One of those is that many of these cells, when they are forming cluster, they have this contact inhibition locomotion dynamics. So basically, that means that kind of inside the cluster, now the cells at the edge of the cluster are trying to polarize away each other. And if they're trying to follow the dynamic of the footprint, so basically they're kind of pulling in opposite direction following this footprint. So we are kind of speculating that could lead to some type of oscillatory behavior in this cluster because you just have this pulling from the different sides of the cluster. And actually, some oscillations of cluster of cells has been observed, but in a different context. So instead of a footprint, was cells that were carrying extracellular matrix, basically. So like they place cells in a gel, in a matrix gel, and basically they observed that over time they start oscillating. So we were wondering that, okay, maybe we can observe something similar in this case.
Hi, I have a question. Great talk. You've shown that how far the cell will get depends on the rate of secretion of the footprint. So if the footprint secretion goes up, the cell can go much further. What happens to RAC then? Does it move? Does it not move? What comes first, the reading of the footprint gradient or the change in the distribution of RAC?
I'm sorry, I'm just trying to remember how this was implemented. I think that the fastest dynamic, so the laydown of the footprint is kind of instantaneous here. So, well, no, not instantaneous, depends on the secretion rate. So you can make it as faster as you want by increasing the secretion rate. And then the dynamic of the rack is always the same in all these cases. It's much faster than how the cell moves, but it's fixed on time. So you can make the laydown of the footprint much faster than actually the rack response, or you can make it slower. Yeah, that's true. I'm trying to think what is going on in the persistent case, if that is the case where rack is faster, no, the footprint is faster than rack. And maybe it is, I don't know, I will need to check this.
Okay, something perhaps to follow up on.
Yeah, I will need to check this. I don't actually remember, but I would guess that probably I would guess that the rack dynamic is still faster than the position of how the footprint is built up at the front of the cell.
All right. So with that, let's thank our speaker once again for a wonderful talk.